Okay, so I wanted to point out to you a few of the big picture items when we're comparing the cell process of mitosis and meiosis. I'm looking at page 82 and 83 in our notebooks. The first thing that I'd like you to do is to read in full the attached worksheet pages 68, front and back, and pay close attention to the details as they go through the actual cell cycle. We're starting up here, we're moving all the way around, and then one of these cells would actually go through this process again. And go through the details here. I will have posted up answer docs for this in the Google Classroom today. The other thing that I would suggest that you do is look real closely at the big picture of this mitosis. Remember, this is making body cells. They start off as a 46 chromosomed cell, and through a simple, well, it's not simple at all actually, but we're making it look very simple. We're taking this diploid body cell, copying everything inside it and including the nucleus. Here we've got replicated chromosomes, and then dividing everything into two finished exact cell copies of the original cell. We call these cells daughter cells, and we also call them diploid because they've got the same number of chromosomes as the original cell, which, like all of your body's original cells, were 46 chromosomes. Then I'd like you to read over very carefully the matched page 155 and 156. 155 is a wonderful overview, and it reminds us the difference between mitosis and meiosis. However, on the back side is really where two very key pieces of information come from. Meiosis, the making of our sex cells, takes one of these diploid cell and through crossing over and independent assortment and two division processes, ends up making 4 cells. So we start off with our cell at rest, then we do our first division, we end up with 2 cells here, and then those 2 cells go through a second division, and we end up with 4. And so how are these 4 different from each other? Why is that happening? And that happens way up here at the beginning during crossing over of prophase 1, when the homologous chromosomes, or 4 sister chromatids, all kind of join up and they crisscross and swap pieces. And then the way that they line up, notice we've got blue on the left side 2 times and then a blue on the right. That's completely arbitrary, that's random. If they lined up with all the blue on one side and all the red on the other, that would be a different set of cells down here at the bottom. But your reading passage here and here not only talks about crossing over an independent assortment, but it tries to illustrate it for you in the imagery. So please do keep that in mind. And again, we'll go over these 2 processes because they look very much like each other. Oops. We've got our starter cell, cell number 7. Then we've got our interphase, which is growth 1, synthesis, growth 2. Very straightforward. Then we've got prophase. Prophase looks a little different in mitosis. There, the chromosomes are just by themselves. They're replicated chromosomes, so they look like X's. The nucleus is dividing, but over here, the nucleus is also dividing, but these chromosomes have matched up with their homologous pair. Remember, we've got 46 chromosomes in all of our body cells, 23 from each of our biological parents. So if this is in your body, in your gonads, making your sperm or your egg cells, that means we're taking all of our parents' genes and we're putting them together. Again, we do a division process. Over here on mitosis, we just have the single replicated sister chromatids pull apart, and then we get new nuclei. Over here, the first division is with those homologous chromosomes, so the pairs divide, but then still you can see they're replicated chromosomes, so they have to divide a second time. They line up in the middle, and the sister chromatids get pulled apart in anaphase 2. And then we end up with 4 cells that are exactly alike. Anyway, happy reviewing. Please do keep in mind the big picture and do a little bit of reviewing before you are back in test mode on Tuesday.